Hello everybody and welcome to this special event in collaboration with the Natural History Museum in London and the Great Exhibition Road Festival. It's great to have you here today. Thank you so much for joining us. Now, for those of our viewers who maybe don't know what Exhibition Road is a main road in London that is a hub for many scientific and cultural institutions, including the Natural History Museum and Imperial College and many other world-famous museums and attractions. And today we are going to be talking a little bit more about Exhibition Road itself, and also speaking to a few of my colleagues here in the Natural History Museum about some of the mysteries of the collection and of the museum, and in fact of Exhibition Road and this area of London itself. Now, if you have any questions at all for us during the show, we are coming to you live, so please do feel free to send in questions for us. We'll be really happy to answer some of the best ones. We'll try and get through as many of them as we can during the show. But I'm not the expert on this topic by any means, so I'm going to introduce you to our guests today. First up, we've got Richard Sabin, who is Principal Curator of Mammals at the Natural History Museum. And joining him today as well is Lorraine Cornish, who is Head of Conservation at the museum as well. Thank you so much, both of you, for joining us today. It's great to have you, and I'm really looking forward to talking to you today, because between you, you have a wealth of experience and so many stories to share about the museum. I'm really looking forward to hearing about it. So thank you very much for joining us today. So let's find out a little bit more about what you do. So Richard, I'm going to start with you, if you don't mind. Could you tell us, I mentioned you're Principal Curator of Mammals, but what does that actually involve? What do you do at the museum on a day-to-day basis?
I'm one of the fortunate people that gets to work directly with the research collections that we have at the museum, the things that people who visit the galleries don't get to see, you know, the 80 million specimens that we have that we use for science, all kinds of science right across the board. I work with the Life Sciences collection, the Mammals collection, and specifically the marine mammals. And there's an image on the screen now that shows me with one of the smaller sperm whale skulls in the collection. Now, there are scientists coming from around the world to use these collections every year, working with our own scientists, and the information that we generate goes out into the galleries, gets published in scientific papers, and actually enriches our knowledge of the natural world. It's a fantastic collection.
It sounds a very exciting place to work, and you know, to be close to so many incredible specimens must be really exciting. And Lorraine, what about yourself as Head of Conservation? What does that mean?
Well, I have the fortunate job of looking after the collections and making sure that they are available for the public to see on display, but also for the researchers. So we look at how deterioration might happen, how we can stop that. I have a team of about 12, and we just look after those collections when we study them, and we make sure that they're in tip-top condition to be able to be used. So it's no 2 days the same. One day I could be working on a dinosaur, the next it could be a housefly. You just don't know.
And maybe some days on the same day you've got all those things as well.
Yes.
Excellent. Well, thank you both for joining us, and I'm really looking forward to chatting with you today. And thank you guys as well again for coming. If you do start sending in your questions, and we'll try and answer them as we go along, because between Richard and Lorraine, I think we've got loads of things we can cover. So let's talk about, first of all, about the collection at the museum. There's a last count, I think there's about 80 million different specimens, huge number, and yet there must be in that collection so much that we still don't know about it. Richard, you know, as a curator, you know, are we discovering new species all the time? Is this an unknown collection that we're still learning as we go along?
There's two aspects here. There's the aspect of we send our scientists out into the field. We explore parts of the world that we still don't know particularly terribly well, and even parts of the world that we think we do, and we discover new things. But there's also the collection, you know, that we have in the museum, most of which was built in the 19th century and through the first half of the 20th century. So much material was collected that we're actually still discovering new species in our collections. And this is the really exciting thing. It's a vast resource, a vast scientific resource. And in fact, we were able to estimate, well, actually not estimate, we did a tally, a total of the number of new species that we described at the museum in the space of a year, and it came to just over 500 new species. Now that's remarkable. That's absolutely remarkable. We think we know the natural world. There's still a huge amount that we've got to learn.
Yeah, that's incredible. 500 species that, you know, this time last year we didn't know, and this is all happening as well in in a year of the pandemic where, you know, we can't travel the way we used to. We're all kind of restricted at home in many ways, and to still have discovered 500 new things to science is quite remarkable. And now, I guess a lot of people might be curious about how you use that collection, because there's one thing to identify what something is, but how do you actually go about using a collection to answer some of these unknown mysteries and questions that you might have? Richard, I'll come to you first.
Yeah, sure. Scientists, students, all kinds of people who have a research interest, they contact us and they make an appointment to spend time working with us in the collections at the museum. Now, you know, when I joined the museum in 1992, which isn't that long ago, but I suppose it is when you think about it, it's almost 30 years. The way that we were using the collections then was still very traditional. And by traditional, I mean that we were still largely just making observations of the outsides of animals, the way they looked, the insides of the animals, the structures that they have, the things that allowed us to classify them within the natural world. We call it taxonomy, the identification of the different groups of plants and animals and minerals and so on, and the categories that they exist within. Now, what's happened over the past 30 years is that very sort of traditional form of identification, examination, and taxonomic study has become much more about what's inside the tissues, inside the materials that form these specimens. So we now routinely look at the DNA that's contained within the bones and the teeth and the fur and the feathers and all the other materials that exist within the collections. It's incredible how all of that has been preserved. And of course, think about the age of the collections going back several centuries. That information about the genetics of these organisms is stored, and it allows us to look and see how things have changed through time. Has there been a loss of genetic diversity in any areas? Not just about the genes, it's also about the diet and the health of the animals. We can take samples from these tissues, and we can look at things called stable isotopes, and these are the elements that get laid down in the tissues of an organism as they feed, as they migrate. It's all recorded. It's like a life history, and it's all there to be studied. And now we do that routinely with the collections these days. It's standard, but the amount of information that we generate is just incredible.
It is, and you know, to think about how it's changed, like you can see in this image, for example, on the screen, that's a very traditional view of the museum of packing the display cabinets full of different things. But of course, it's not just now understanding what those things are or where they came from, but we can unlock a tremendous amount about them just using new technology. Lorraine, I imagine as someone who works with such a variety of things, you must have had some strange questions about the conditions things are in, or how they ended up coming into the museum, I'm sure.
No, absolutely. Such a diverse collection. There are many unsolved mysteries, which is part of the fascination of working on them, of course. For me and for my team, we're trying to understand what causes material to deteriorate. We call them agents of deterioration. So things like too much light, too much humidity, too high a temperature. And we study these methods and try and understand that. So, for instance, in our Earth Science department, we have many beautiful fossils that are made of a material called pyrite, often called fool's gold. And it was discovered that these specimens would just deteriorate. They'd go to a white or yellow powder. No one really knew why. And so one of the early bits of research that I did with people when I first joined the museum was we're trying to understand that mechanism, look at the chemistry and understand it. And we discovered that, in fact, it's oxygen and it's high levels of humidity that trigger this event. And so now we try and make sure we store our specimens in a great environment, and we will put some of them in literally in bags. They look like crisp bags. We put oxygen scavengers in, and then we store them that way. So we're really taking and reducing that oxygen level. So trying to solve some of those mysteries is a fascination for us. And then we learn more about the collection and the specimens as we go. Then we'll publish that information, because then that prompts other people and other scientists to look at the same sorts of problems in different ways. And then we can sometimes come up with an even better result and a way to actually preserve the collection. So it's a very collaborative piece of work that we do. in the global community. And it's really important that we publish on that sort of thing too.
Fantastic. Yeah, it's a bit like being a detective or something when you're trying to figure out what's happened to this specimen that we maybe haven't looked at for 50 or 100 years, and it's like, trying to learn that process and then apply it to other things so that they're going to be here for future generations. It's a fascinating process. And we've had lots of questions coming in, guys. Thank you so much for these. Let's tackle a few of them. So I'll throw these questions out to both of you, Rich and Lorraine, and just chip in if you have an answer. So the first one here comes from Fadia. They're asking, what is your favorite thing in the museum? That's quite a big, I guess that's like, how do you pick your favorite child, surely?
It's just like that, actually. Sometimes I have two sons, and like I couldn't say which. They're both wonderful in different ways, You're
not going to say it on air, Lorraine. That's the key thing, isn't it?
Yeah, I think sometimes it's down to just being fascinated by how mysterious a specimen is, or what you don't know and you want to know more. Others, it's working on a specimen. So Richard and I both worked for a number of years on bringing hope into Hinsey Hall, and suspending her there for people to see. And that was a work of love and a lot of science involved. But I think we both feel quite strongly about hope. In fact, don't we, Richard?
Absolutely do. Yeah. And although I dearly love hope, my favourite specimen, believe it or not, is the skull of it's a species known as the rough-toothed dolphin, and it's completely decorated. It's been inscribed and scrimshawed, decorated across its entire surface, and then coloured. Now, this would have happened on a ship a couple of 100 years ago. A sailor, with nothing better to do, would have gotten hold of the skull and then would have used all the available materials like sewing needles and soot and any other pigments that were available to create pictures of his life and his travels on the surface of this skull. It's just remarkable.
Great, a good question there, Fadia. It's always a tough one to pick your favourite out of. My favourite's the giant squid in the Spirit collection. That holds a special place in my heart. I remember going on a tour when I was still a university student and saw that giant squid, and I was like, I really want to work here. And years later, I was doing those tours myself. So it's got a special place in my heart. Another one here, this is a question from Malika, referring back to when we were talking about new species. How often do you find new things? I guess that could be either in the collection or possibly out in the wild. Let's maybe look at the collection, since that's our sort of focus today. How often do we find new things?
It's not uncommon. It's not uncommon for that to happen. There's work going on all the time. There's constant revision going on of what we've done before, and the assumptions that we've made, and the identifications that we've made. There's a constant recalibration, if you like, a resetting of our knowledge in some areas. And in some cases, we do find entirely new species, and they've been kind of masquerading as something else. There was a case recently that it actually happened in the USA. A new species of whale was described. And this has been known about for decades, but actually hadn't been properly identified because there wasn't enough information. So, species identification, sometimes things can come along that are just entirely different to anything you've ever seen before, look obviously very different. And then others look incredibly similar, and it's about what's actually going on inside. Those are the things that we need to look at in detail. But, more often than you think, keep an eye on the museum's data portal and the publications that our scientists are producing, and you can see month by month new species being described. It's incredible.
Brilliant. And it's really exciting to think that, we think we know the natural world, and actually that's not, there's probably more for us to discover than we realize, which is, again, why we need to protect it as well. Lovely. There's so many questions coming, guys. It's really great to see them. Let's try. I don't think I'm going to get through them all, I'm afraid, but we'll try and pick out a few of them. This question on Facebook from Aruna. This is for you, Lorraine. Can you share any one of your challenging field expeditions? So is that something you get to do? Do you get to go on field work?
I have done in the past. One challenging field expedition was when we worked in Gibraltar and we were looking for Neanderthals. And one of the sites was down several hundred steps off of a cliff. And then there were these fantastic sea caves. Okay. And we were excavating within these caves. And then there was another beach nearby, which also had other caves, but we didn't understand why there was a lot of barbed wire and we couldn't get there. Then we found out that actually it's where they do practicing explosions, and then they have like army sorties going in. And so then we realized every time we would hear this noise next door, that was what was happening. So that was a bit disconcerting. And one day a whole load of soldiers came up in a boat, and off popped obviously the colonel or someone, and there were several crates of champagne. It was his birthday that day, and he thought we might just want to have a little toast on the beach with him. So there we were, all in our field gear, sieves, you know, drinking a glass of champagne to wish him a happy birthday. And so what we did was we just said that actually these buckets are full of sand and everything. They're quite heavy. Do you think some of the soldiers could help us? So we actually got some of the squaddies to help us do some sieving in the sea. So it was quite unusual, I have to say.
And I bet they never thought they'd be doing that. You know,
No.
not their typical daily duties, is it? No. Excellent. Thanks very much for that question, Aruna. This is another interesting one from Julia on YouTube that I haven't actually thought about before. It's like, how do you start or complete a collection? Do you ever finish a collection? I guess it feels to me like, well, how would you know when it's finished? I suppose is the sort of follow-up, isn't it? I don't know, Richard, have you got any thoughts on that?
Yeah, it's like I suppose if you were to travel to an entirely unvisited, if you like, part of the world, you go to a part of the world, a tiny island, you've never been there before. What's your objective? Why are you going there? Why do you want to set foot on the place in the first place? You know, what do you want to learn? So you need to have a plan. And of course, centuries ago, it wasn't about having plans for scientific discovery necessarily. It was more about curiosity. There were, unfortunately, largely gentlemen travelers, but actually, as we're discovering more and more women who were also traveling, who were collecting and were describing and, you know, starting to have an appreciation for what they were finding. Scientific specimens, natural history. They were looking at the cultural artifacts. They were looking at a whole range of things, but there wasn't a sort of a formalized way of studying and presenting these. Not in the same way that we have today. When we go anywhere today, we go with a purpose. You know, we go with a sense of what is it we're trying to achieve. We are often, of course, now, when we travel to other parts of the world, being invited by those countries to go there. We have all of the ethical considerations about what we collect and how we collect, and making sure that we don't impact on the environment, on the plants and the animals that live there. Do we ever finish collecting? Of course we don't. We never do. Collections.
No good collector ever does. You know, whatever it is you're collecting, you want to make that collection bigger and then move house.
Yeah. And collections expand. You know, you can have a collection of 50 specimens from a particular area, and scientists will come along and look at that collection, and they'll extract things, and things will be subdivided, and you know, it's growing all the time. It's not just about the physical specimen itself, but it's also the data that's generated from that specimen.
And also Excellent. We get collections from collectors in the past. So we've been donated a collection, you know, Charles Darwin or Wallace. And so that those collectors, we have collections from them, and then we have to kind of keep it that way, but also integrate them into the larger collection. So their collections come and go, but it's an ever increasing collection that we'll have, and that's what we have to plan for. If we have more collections, do we have enough space for them? So we have to think about that very carefully when we're going to do any field collecting or receive any collections.
Yeah, of course. Yeah, we've got to have somewhere to put it and get back. Yeah,
absolutely.
Yeah, no, it's fascinating to hear that, and interesting to hear. You know, you touched on it, Richard, a little bit about the ethics of collecting as well, and how we want to build the collection, but of course we've got to consider why are we doing it, what and what it's important that there's a reason behind it. We had a question come through on YouTube asking about that. Now the next question has actually come in from Sue on YouTube, and actually leads quite nicely, I think, on to the next segment I want to talk about. Sue's asking, with new technology coming in all the time, do you then go back to preserve mammals and use this new technology and find out new things that you didn't know before? And I think a really nice example to illustrate this is, of course, Hope, the blue whale. And we've mentioned Hope. Anyone that's been to the museum in the last couple of years can't have missed this beautiful specimen hanging in the main hall. But if you haven't visited the museum in person, This is what it looked like. Remember when people could gather like that under the blue whale? Those were the days. So yeah, Richard, I know both of you have worked very closely on this, and we'll talk about that. But I know, Richard, in particular, you have done a tremendous amount of research into this individual specimen. So it's not just identifying what it is, of course, but there's a lot more to it than that. Can you tell us a little bit about that?
Yeah, there's a lot to say, but I'll try and keep it brief. In terms of what we wanted to achieve with Hope, it was about moving her from where she was in the old Mammals Hall, called the Whale Hall, and bringing her into Hinsey Hall to replace Dippy. We wanted to have her in a really dynamic diving pose, and so I was lucky enough to spend time in the field in the Pacific making observations of blue whales feeding. And I brought those observations back and was able to put those into the pose, if you like, of Hope that you see there. But it was more than just that. That's an illustration of the science of the behavior, the feeding behavior of these animals, which is relatively new information. But we started to look at what's inside Hope's bones, if you like, and also her baleen. These are the plates, the sheets of keratin, the protein that hangs down inside the mouths of filter feeding whales that they use to strain their food from the ocean. And the great thing about that material is that it lays down a lot of information about the animal's life, about its health, about its movements, about a whole range of things. So the first thing we did, a group of us at the museum got together and we worked with a laboratory and colleagues outside the museum. We took samples from Hope's plates of baleen, and we were able to look at things called stable isotopes. And I mentioned these before. These are the elements which are laid down in our tissues when we feed, when we drink, when we move around the planet. There are different types and different levels of these elements which are laid down, and we can read them, we can decipher them, and we can actually see to a certain extent what's been going on. And with Hope, we're able to determine that in the last few years of her life, based on the amount of growth in those plates of baleen, that she'd been moving between the waters in the north off Iceland, Greenland, and then heading south past Ireland and the British Isles down to the west coast of Africa every year, moving backwards and forwards, feeding, breeding, backwards and forwards. But then something happened. She stayed a lot longer in those southern warmer waters, and we were trying to understand why that was. We carried out a second series of tests, again using the baleen, looking at hormones. Now, hormones, stress hormones, pregnancy hormones, all those things that your body is producing get laid down in your tissues, and we're able to extract from Hope's baleen information that told us she'd been pregnant. In the last 2 years of her life, she'd been pregnant, she'd gone full term. It looks as though she'd given birth, and she spent those months in the warmer waters further south, nursing that calf. Now, what incredible information that is. That's the kind of level of investigation that we make of our specimens, and that's just scratching the surface, literally. It's routine, but it gives you such an amazing amount of background detail to add. And also the important thing is that Hope is a North Atlantic blue whale. It's a subspecies of blue whale that was heavily impacted by commercial whaling in the 19th and early 20th century. So we actually lost a lot of these animals before we had a chance to study them. But with this data that we're getting from specimens like Hope, we're adding to our knowledge of how these animals used to feed and the breeding intervals, and comparing that with the modern day. And it helps to do things like conservation planning, have an awareness about the things that actually impact these species. So it's a really good thing that we're doing.
It's incredible, the amount of detail that we're now able to extract from a specimen like that. When that specimen first arrived in the museum, as you say, it was exciting to have it just as an example of this incredible species. Those people could have had no idea that we would have been able to tell them now, where it had been, the fact that it was pregnant, and all of that, and getting really precise data on it is really exciting, really incredible. Yeah. And Lorraine, I know you've worked very closely with Richard on Hope as well, and you've been unraveling a few mysteries about this specimen yourself, haven't you?
Yeah, and I think actually one of the mysteries that we were able to, which was very interesting to us, was when we first took Hope, down from where she was originally displayed in our mammal hall, because as we took her apart, as you can see, I'm on the scaffolding now, taking her mandibles apart, we learned more about how she'd actually been put together. So in fact, on her skull, in her palate, we actually found the original signatures of some of the team that had actually put Hope up in the 1930s. Between her vertebra, we found old newspapers between 1932 and 1934 that they'd obviously used to pack out. So it's really fascinating to be able to reconstruct how she'd been put together and all those years ago. And even now we're analysing, in fact, we're doing some analysis on her to the first evaluation of how fungal communities are actually inhabiting her surface, really, really tiny little fungal communities. And are they having any impact in terms of deterioration? So there's some active research going on there. So we really enjoy that the specimen never ends in terms of the information it's giving us, but it's also for us to learn about deterioration processes and how we can preserve her for the future as well.
Fantastic. Yeah, no, it really, again, exciting to think. I'd never even imagined that we could have found this sort of thing out or been exploring one specimen to that degree of detail. It's a lot. You're not just glancing at it, you're really getting right down to the microscopic. It's a lot of work, but it must be really exciting to be involved in that. I'm going to take a quick pause here and look at some more questions from our viewers. Thank you very much, guys, for sending them in and waiting as we work our way through them. We've got lots of people been asking broadly about the differences between collecting and looking after collections in the past versus today. I guess we've sort of touched on that already a little bit. I guess, there's a big difference between, I guess in the early days, collecting was probably quite indiscriminate. I guess there was possibly an element of kind of stamp collecting to it. You want to get one of everything and say you've got it. Richard, I think you touched on the fact that nowadays collecting is very much driven by sort of research needs and sort of strong ethical arguments. So yeah. I don't know if we want to touch on that a little bit for viewers. I think they're quite interested in those kind of past-present differences.
Yeah, you're right. I mean, there was a lot of relatively indiscriminate collecting that went on in the 19th century. We can't make apologies for it. The collections exist. They were very much of their time. The huge numbers of specimens that were collected. Now, there are some who would argue that those early collecting expeditions had a detrimental effect on the numbers of species in the wild. That's debatable. I think the thing that's really had the effect on the numbers of species in the wild are our activities as a species, as a human species. So, the effects of the Anthropocene with the pollution and the development and a whole range of things which have really reduced the numbers of species over time. But the way that the material was collected and the way that it was preserved back then, actually, I think we were lucky to a certain extent because the techniques that they were using for preservation allowed a certain amount of stability, and Lorraine can expand on this, but the way that the tissues in these animals that were collected a couple of hundred years ago have been preserved, and I think the luck that we've had in a lot of cases with our old museum buildings has meant that we can revisit those collections and we can find out this new information. It's an ethical consideration. In a lot of cases, there are places that we visit and that we can sample from nowadays, but the samples that we take are not whole animals anymore. They're environmental DNA samples. We collect water from a pool where we know that animals go to drink, and then we can actually look at the DNA that's present in that water and see all of those different species. There are ways that we can get information from the wild nowadays that are much more sympathetic, if you like, and much more appropriate. We have an awareness now about conservation. We have an awareness now about the need to preserve the natural world that just wasn't around back in the 19th century. There were a few individuals that had that foresight and knew that things were starting to change. Our very own director at the Natural History Museum at the end of the 19th century made a public comment saying, we need to keep an eye on what's going on because there seem to be things happening on the continent of Africa and across Asia with numbers of animals reducing, less frequently being seen. They knew that something was starting to happen. So collections have become much more important to us in terms of understanding that we have a great responsibility to preserve these things. In the 19th century, a lot of stuff was considered to be disposable. In fact, we did lose a lot, didn't we, Lorraine?
Yeah, no, absolutely. And I think the thing about collecting now is, certainly before we go and collect, we do a collections impact statement. We have to go through a set of criteria to almost justify what questions are we trying to answer, what impact is that going to have, but also just if we bring these things back, what are we doing? How are we benefiting science? How are we, talking to the public about it? So it's much more considered. And I think, now we're in a world of digitization and much more global integration. Sometimes you're not collecting as much, and you can, as Richard said, you can get data without actually physically collecting the specimen. You can capture information in different ways. We're always looking at how we can do that better, and maximize the amount of information we can get. But it's also about preserving our specimens, making sure we're almost future-proofing them. So not trying to do any treatments that might hinder any future analysis that might take place. So we're trying to guess almost what might happen in the future in terms of technology. So less is more, I think, from my perspective, in terms of conserving the collections. We just try and do the minimal, but just to make sure that they're preserved and they're available for study.
Fantastic. No, really, really great answers. Thank you very much, guys, for that. And I think just following on from that very, very quickly, I'll ask each of you a quick fire response here. Evie is asking from YouTube, if there's one place in the world that you could have unlimited access to explore, where would it be and why? So let's, for a moment, let's put all the ethical concerns aside, and just for fun, if you could have unlimited access somewhere in the world, is there a place or an environment you want to go and collect to your heart's content?
Yeah, I don't know about collecting to my heart's content, but certainly making observations. I've got a great passion to go to Greenland. It's a vast island continent almost. It's an amazing place. But the thing about Greenland is, we know there's a huge amount of diversity in the species that are associated with that country. But it's changing, and it's changing rapidly. The glaciers are melting. Temperatures are rising. Things are happening, and they're happening very, very quickly. There are observations that are being made. There's scientific data which is being collected by the Greenlanders themselves and the scientific institutions that there are on Greenland. But for me personally, I want to see these great wilderness spaces before they're gone for good. And there's a very real danger that may be the case. We're doing an awful lot to try and stop that from happening. But Greenland for me would be the kind of the go-to.
Great choice, I think. I'd love to go there as well. And Lorraine, what about yourself? Is there anywhere that you?
I've got two places probably. If money were no object and science had moved on, obviously I'm so fascinated by the mission to Mars and the collecting that's going on there. And the museum's been a little bit involved in some of that. So if money were no object, that would be fantastic, wouldn't it? But also, I think I'm a dinosaur girl too, and so I would love to go out to somewhere like Wyoming, not necessarily to collect, but I'd love to go and see these amazing fossils being excavated, but just to understand the landscape and to talk to some of the scientists out there. So that would be my dream too.
Fantastic. Yeah, great choices. And I second you, Lorraine, on going to Mars. I think that'd be good. And if you guys are interested in Mars, we were just talking about the Perseverance mission the other day. You can check out our video on our channels if you want to see that. But yeah, I definitely would want to do that, I think. Brilliant. Well, let's move on a little bit and talk. I want to talk about, we've talked quite a bit about specimens and hope, but one other aspect of the collection that's a little bit different is the use of models. Which sometimes we've got real specimens, but sometimes we've got these sometimes very detailed, accurate models of organisms that have been used both in display and also in the research. And there's one above all else that I think really stands out and you can't miss it. And it's this, you can see it in the image here, the blue whale, which is in our whale hall, a stunning model. Now, there's a bit of mystery behind this because this is a very old model. And just the question of how on earth did something like that get built, because you've got, I guess, two issues. One is physically actually building it, but the other is getting it as accurate as possible based on the technology you had at the time. Now, Richard, I know this is something that you're personally very interested in. Can you tell us a little bit about the story behind this model? Because it seems like a bit of a mystery as to how it got built in the first place.
Yeah, bear in mind that the Natural History Museum at South Kensington opened in 1881, and our first director, Richard Owen, he wanted blue whales to be part of the spectacle, part of the public galleries, and actually that was why Hope's skeleton was collected, but we didn't have the technical ability to put it on display back then. The use of models became more and more a part of early kind of museum galleries in the late 19th century, because they realized that there were things that they couldn't collect from the wild, preserve as taxidermy in the same way that you might see a taxidermy gazelle or a taxidermy lion. You can't do that with whales and dolphins, and particularly not with the largest animal that's ever lived on the planet, which is the blue whale. So we were very fortunate to have working for us a man called Percy Stanwitz, who was the museum's head taxidermist and model maker. And Percy, very much like I did in a way, he spent time in 1914, 1915 down in the Antarctic with the whaling fleets, and he was making observations of blue whales and fin whales, but particularly the blues, because he was pushing very hard for this life-size model of a blue whale to be built in the museum at South Kensington. He wanted to see it in the whale hall with the skeletons that were already there. You know, the blue whale skeleton, Hope's skeleton was already in place, plus 4 other huge skeletons that were in place. He wanted this. He wanted the visitors to the museum to see how enormous these things are. And so he got permission and he set to work. It was in 1937, early 1937. It took just over a year to complete, set to work building a life-size model of a blue whale. It was constructed from, as you can see, strips of timber, a wooden frame, built a little bit like a ship, if you like. The timber was laid over the top of the main frame, and then it was covered in layers of effectively chicken wire, you know, this fine wire mesh. And on top of that, they placed layers and layers of plaster. And the plaster was then sculpted to show all of the detail, the external detail of the blue whale, and finally painted in the colors that we see with blue whales. And the incredible thing is that this model was actually suspended as it was being painted. And one of the things that they reported was the gentle rocking motion of the blue whale model that was caused by the people painting the surface actually made them feel seasick, which I think is kind of ironic in a way. But what we ended up with was this certainly life-size model of a blue whale. This design was then sold to other museums in the world. This was the first time any museum in the world had done this. We sold the idea to the American Museum of Natural History in New York, a fantastic museum. And of course, our colleagues in the USA decided to make their own model, which they made slightly bigger than the one at the Natural History Museum for obvious reasons. But
of course, yeah.
The accuracy of the specimen of the model, how accurate is it? Well, bear in mind that the dimensions, the measurements, the external characteristics were all based on not underwater photography or you know, scuba diving, because that wasn't a thing until the 1960s and 70s. We didn't have detailed underwater footage of these animals in the wild. It was based on stranded animals. It was based on whales which were taken to the whaling stations that Percy Stanwitz saw in the 1914-1915 period. And it was based on lots of statistics which had been taken by the people, the scientists working at the whaling stations. And so what you have with our model is a slightly distorted, slightly bloated version of a blue whale, because what we now know from the wonderful programs that are made by, say, the BBC, Blue Planet, you see just how streamlined these whales are. And there was a famous American scientist a few years ago who said that as much as he loved the Natural History Museum's blue whale model, if it was an actual living animal and it was found washed up on a beach, it would probably need to be described as an entirely new species. So maybe not so accurate.
Yeah, it's certainly a striking model, and yeah, interesting to see how the appearance of it was impacted by basically the only way that Percy was able to get access to it easily. It sounds like it's high time to get a new one made.
Controversial.
Now, Lorraine, as someone who you obviously work with specimens. Do you work with models like this too?
I work with everything, Alastair. So specimens, models, books, illustrations. We work with them all, and in the museum we have a fascinating collection of models. some beautiful historical models that kind of, as Richard said, it's a snapshot in time of how things were interpreted at that particular period. So as well as the blue whale model, we have an amazing collection of glass models made by father and son team, the Blashkas, from the 1850s. And these are amazing. And the collection we have is a lot of sea creatures. So they would have shown people who had never normally seen these creatures what they look like. Very, very fragile. We've got a lot of dinosaur models, fish models, beautiful insect wax models made by this amazing woman called Grace Edwards in the early 1900s. Used as teaching, used as also to teach people, so on display in the galleries, but really to interpret how science was at that stage in terms of what this animal might have looked like. And certainly the dinosaur models are really interesting because some of those models now are known to not be accurate, but it's just a snapshot in time in terms of how people saw those animals in the day.
Yeah, and actually, while we're on that topic, Lorraine, about models and the accuracy of them, I wonder if we could talk about the iguanodon, because that's it's a dinosaur, as most people will know, but it's gone through a little bit of a transformation in how it looks, hasn't it? Because obviously part of it's based on limited understanding.
It is, and I think so it's very interesting for those people who've ever been to our beautiful Crystal Palace Park in South London. Was one of the first ever displays of dinosaur models and how they might have looked. And here we've got a couple of pictures of an iguanodon, and these show how they thought in the day that the iguanodon had a little horn on its nose. And here we've got two iguanodons actually looking at how those were perceived in the day in the sort of 1850s. You've got one at the back there on all fours, so looking like an elephant. So that's one interpretation about how these creatures would have moved around. And then in the foreground, you've got another one sort of lounging like lizard-like on a cycad. And that just reflects, in fact, what the idea was back in the day. And we have some scale models of these dinosaurs in our collections. And as they found more and more pieces of iguanodon, shall we say, now been renamed as Mantellosaurus, they realized that this horn core was actually a thumb spike. So as they discovered and excavated more and more of this dinosaur, and there was a massive collection of iguanodons found in Belgium, they pieced together this and they realized that there was a thumb spike. So even our own Mantellosaurus dinosaur that we've got in the museum galleries, it's on display in Hinsey Hall, that underwent a transformation as well. At one stage, it was a bit like an upright sort of kangaroo rabbit look. But as they discovered more and more about these dinosaurs, they realized the back legs were really strong. The forelegs were very slender. They would have had a really long tail. So in fact, the whole posture has been changed. And so I was lucky enough to work on a project to actually transform what you can see now on the left-hand side into now what we understand as the proper anatomy for these dinosaurs, which we've got displayed in Hinsey Hall. A lovely piece of work and really challenging, but really interesting to learn more and more about the science of these animals as you go along. And there is our beautiful Mantellosaurus from the Isle of Wight on display in Hinsey Hall.
What a fascinating journey that's been on, because you look back at those models from the Victorian era in Crystal Palace Park, which look like giant lizards, and then to something like this, which looks far more elegant and athletic. It's quite a transformation. But that, I guess, that just goes to illustrate how one thing what's so interesting about models, because they don't change once the model's made. That's your kind of time capsule of what we thought, and now we can bring it forward using new information, new science and technology. That's fantastic. Thank you very much, guys. A couple more questions coming in. This question comes from Stavros on YouTube. I think I know the answer to this one. Is the museum space adequate enough for all the exhibits? I'm going to say it's a massive no.
No, we only have a very tiny percentage around 27,000 items on display in our museum in South Kensington and Tring, and we would love to put some more out on display. We have touring exhibitions as well, of course. So one of the projects that we're involved in over the next 5 years actually is how to unlock some of the gallery spaces that we aren't using for the public at the moment, and we're actively looking at how we can do that so that we can actually increase our gallery space for the public to come. So never enough space for the collections to be stored, let alone displayed.
No, yeah, I can imagine it's going to be an ongoing thing. As long as the museum exists, this is going to be a challenge for it as the collection grows. Excellent. Well, we're rapidly running out of time. It was so much to cover, but it's been lovely having all your questions, guys. So thank you very much for that. Before we wrap up, we did mention this actually very briefly earlier on in the show. Obviously, with the museum currently being shut, this wonderful collection is not as accessible in the way that it normally is. And of course, there'll be not only scientists and researchers all over the world, trying to answer their own questions about the natural world, but the general public as well. But the museum is actually in the midst of a project that's making as much of this collection as possible available to people virtually. Could you tell us a little bit about the data portal and how that works? Because I can only imagine that this project is going to reveal more and more about the secrets of this collection in the natural world.
Absolutely. The drive for the museum over the past almost decade now has been to digitize our collections, photograph them, 3D image them, make sure that the collections data that is associated with them is put onto a public accessible platform. This is the Natural History Museum data portal. You can search our collections. At the moment, we've got, as you can see, around about 10.8 million records in there. And that comprises almost 5 million of the museum's 80 million specimens. You can search for the species that you're interested in. You can search for the time period, for the geographical location. It's a huge amount of information. There are photographs. We're digitizing specimens in 3 dimensions. One of the things that Lorraine and I did when we moved Hope's skeleton was to do a 3D scan of her, and we created a computer model which is available on the museum's website. We're trying to get as much information out into the public domain as possible, so people, regardless of who you are and where you are and what you're doing, can have access to it.
Fantastic. Well, that's great to know, and hopefully, I'm sure some of you guys watching today will go and check it out and have a little rummage around through the virtual collection. I'm sure you'll find something of interest in there. But we've run out of time today, Richard and Lorraine, but it's been lovely hearing from you. I knew there was going to be some great stories in there, and it's been really great of you to give up your busy days to come and talk with me and to talk to all of our viewers and answer their questions. So thank you both very much for joining us, and all the best with your work, and hopefully we've got plenty more to talk about in another meetup in the future, I'm sure.
Thanks. It was a pleasure, Alastair. A pleasure.
Thank you both. Right then, well, thank you very much, guys, for tuning in. Sorry if we weren't able to answer all of your questions. There was lots of them coming through there, but hopefully that's given you a bit of a flavour behind the scenes at the museum and some of the many mysteries and questions that our scientists are working on. It's been lovely chatting to you all today. If you enjoyed the show, please do let us know in the comments. And there will also be a link to a survey that you can feed back and let us know what you think, because we love doing these shows and we want to keep doing them. So do let us know what you think in the comments below. But thank you so much for joining us today. And as a reminder, this has been a great experience to work with the Great Exhibition Road Festival, which has a whole series of online events that you can follow. So please do check that out if you get the chance. But thank you so much for joining us today, and we hope you enjoy the rest of your day. Thank you.